So let's briefly, what I want to do today is I want to review kind of where we are and kind of put everything in context. And then I want to talk about, there are many techniques that you would use in endocrinology and studying hormones and how they work. But I want to talk to you about two critical sort of techniques that sort of revolutionize the field. And they aren't necessarily used just in endocrinology, but you'll see, and they're old school techniques that are still used. You'll still read about them because they're still stated true. The radioimmunoassay, and the radioreceptor assay. Here are the basics of it. Remember, you have a midterm a week from today. Here's basically where we're at. We learned that, clearly important, because I've said it over and over again, a hormone is a compound that's synthesized, secreted, travels traditionally in the blood. It has to travel in the general circulation to be a true endocrine hormone. It has a receptor. After binding, it produces some action, and it is degraded. You should be able to talk to me about this definition, and you should be able to talk to me within that about endocrine and paracrine and autocrine and intracrine hormones, because the field's broadened such over the years. Then I gave you a boring list of hormones that we're all going to put into context, but most of the hormones we're going to talk about in the next section. And if you guys still complain I go too fast too, so if I'm going too fast, stop me. Maybe you just practice saying slow down just so you get into practice. Many of the hormones that we'll talk about in the next section of the course, reproduction and development and growth, are hormones that are part of one of these hormonal axes. Not all hormones are, right? Insulin, glucagon, calcitonin, oxytocin. Those hormones aren't a part of the axis, but many of the hormones we'll talk about in the next section are part of a hormonal axis. And a formal hormonal axis involves the hypothalamus, with release or inhibiting hormones that regulate the anterior pituitary, also known as the adenohypophysis. Lisa reminded me, I didn't point out, it's also known as the pars distalis. I'm going to call it the anterior pituitary. The posterior pituitary is the pars nervosa. You'll read those terms in different places. I'm going to call it the anterior pituitary. Then there's some peripheral gland that in a real endocrine axis, in a classic endocrine axis, will have negative feedback. And I've given you at least two examples of when that negative feedback can be important. The use of estradiol and progesterone in birth control pill. Birth control pill works. Or the hormone IUD or the patch or whatever you might be using. It works because of negative feedback. The other example I gave you is an absence of negative feedback that leads to hypertrophy. In the example of the goiter. And then we talked about one that's not a real axis. Because it doesn't really have a peripheral gland, a peripheral hormone, doesn't really have negative feedback, doesn't have a releasing hormone. Almost an axis. Growth hormone, which has a releasing hormone and an inhibiting hormone, but to my knowledge, no negative feedback. If you find a paper that says something different, bring it to me, let's talk about it. We'll discuss it in session. I've not been convinced. Your book says there's IGF-1 negative feedback. I don't know the evidence for it. Then we talked about a real axis, the hypothalamo pituitary adrenal axis. We talked about a more complicated one. I'm going to draw it like this for now. It's going to get even more complicated on Thursday. On Tuesday. 2 Tuesdays from now. There's even more stuff going on. The hypothalamo pituitary gonadal axis. And the hypothalamo pituitary thyroid axis. We talked about how GHRH uses a G-alpha stimulatory, GHIH uses a G-alpha inhibitory, GnRH and TRH are using this GQ different system. Growth hormone is using this JAK-STAT second messenger system. ACTH, LH, FSH, and TSH are all using a G-alpha stimulatory, which can affect the physiology of the cell, actually in all these cases, as well as transcription translation. IGF-1 has a special mechanism with this tyrosine kinase, and all the steroids work by these, and the thyroid hormone, by these zinc finger proteins that regulate transcription. There are other things that steroids do too that we'll get into later in the course. Oh, one other thing I want to point out that has confused people in previous years is the vertical arrows and stops. The vertical arrows and stops means that the hormone above is affecting the hormone below. Doesn't mean that it's being converted. Whereas the horizontal arrow, T to E, means that T is being converted into E2, T4 is being converted into T3. So don't be, people have been confused by that in the past. The vertical arrows don't mean that TRH is turning into TSH. The TRH still is stimulating TSH, not being converted into it. Yeah. Are you dead?
Can you repeat one more time which G units are involved?
So GHRH is a G stimulatory, GHH is a G inhibitory, CRH is a G stimulatory, a G sub S, GnRH and TRH work by this GQ, and I don't know what the Q stands for. Or I think it's called Q/11 or something. I have no idea what that stands for. So what we've been talking about now is how you measure, how you determine that these things are hormones. Because each one of those hormones in that pathway, or in that cascade, in that axis, has to be a hormone. You should be able to talk to me about hypothalamic hormones and how they aren't found in the general circulation and generally wouldn't be considered endocrine hormones, but rather paracrine hormones. But for each one of those compounds to be called a hormone, it has to meet this criteria. You know that if it's a protein or a peptide hormone, it's transcribed and translated. You should know how to use RT-PCR to measure transcription at each one of those, for each one of those hormones. You know that for the steroids and thyroid hormones, they're synthesized biochemically, so you can't look for genes for those hormones. You should know, well, you'll learn today how to determine if they're secreted and travel, and you'll learn how to measure their receptor binding. That's what we're going to do today. But you should know how to measure their action. We talked briefly, and you'll get more techniques, especially in your discussion section. So you'll review a paper on Friday that'll talk to you about how people figured out which second messenger system is being used. So you get a paper today that kind of picks that apart. But I also want you to keep in mind as we approach the midterm. Am I going too fast? Or am I good? I'm going too fast. Maybe I should put some other tea in my cup. So down a little bit. As you approach the midterm, don't forget that before all the fancy techniques, including the one I'm talking about today, which was developed in the 40s or something, the way that these hormones were discovered was by good old classical biology and bioassays. You take something out and you see what happens. Put something in and you see what happens. That's how hormones were discovered. You suck some blood out and people found out that there was something in the blood of females that if you injected into a young female would cause the uterus to grow. That's how estrogen was discovered. Or people would take organs out, inject them into another animal, see what happens. Remember I told you thyroid hormone was important. Remember I told you, I had a student once who was kind of early office hours. She didn't do so well in the first midterm. But after that, she aced it. I said, oh, what happened? She showed me her notes. She used to make little notes in the margin. She said, you make faces to say things when you say things that are important, so I can predict what's going to be in the midterm. So if I say, this is important, and I've mentioned something more than one time, you should think about it. This is important. I told you that thyroid hormone was discovered because some guy named Gudersnach chopped a horse up and fed parts to tadpole and discovered that the thyroid gland made tadpoles metamorphose and turn into frogs. So there was good old biology of where you take things out and you see what happens, you put things in. The pituitary was called the master gland because if you take the pituitary out, your metabolism gets screwed up. You don't grow. You can't reproduce. You don't have a stress response. So even though we now know the pituitary is controlled by the hypothalamus, it was thought of as the master gland because it seemed like it controlled everything. Everything just went all to Hades if you took the pituitary out. You should know how to do experiments like that and how to generate a proper control. You can't just take something out, you have to have a negative control. In some cases, you have to have a positive control. And those are things that you're also doing in your discussion section. So that's the way you should be thinking about as you approach my exam. How would you design an experiment? What tests would you use? What techniques would you use? And you have a small battery now that's going to develop as we go into the course. So a key, other than just pulling things out and seeing what happened, a key to endocrinology was the development of something called the radioimmunoassay. I'm going to tell you in detail how that works and we're not going to do it today. What's your name? Sharon. That's what I was going to say. It helps if you guys sit in the same spot. Yes?
Before you go on to the assays, I had a clarifying question about the definition of the hormone. So when we went over it the first time, we broke it up into steroid and peptide.
Yeah. Well, there's no difference between a steroid hormone and a protein hormone other than how it's synthesized and how it does the stuff. It still meets this functional definition.
Right, but I was just wondering if you could elaborate on the difference between its actions, like the slow and fast.
So the idea was, at least traditionally, the idea was that steroids had slow action, meaning that a steroid has to go into the cell, bind its receptor, bind to the DNA, and upregulate a gene, which then has to be translated. So you don't see an effect on that cell or on physiology for quite some time, typically, right? So if I inject a young woman with estrogen, you don't immediately grow the lining of your uterus. Whereas something like epinephrine or insulin, it's an immediate response through second messengers. If I inject you with insulin, I should stop saying this, but I could actually kill you by injecting you with insulin. All of your glucose would get sucked up and you go into a hypoglycemic coma. It's an immediate fast action. And so it was thought that proteins worked only through these second messenger systems that caused fast actions that changed the physiology of the cell, as opposed to steroids. So we now know that many of those hormones, although they do have these fast actions through second messengers, these amplified signals, they can also, through those signals, affect transcription. You can, through G-alpha stimulatory and G-alpha, the Q one, through the PKA or the PKC pathway, some of those kinases in the cascade actually affect transcription factors that then can change gene expression. Growth hormone is a classic example of one that, am I going fast? That does have fast action by affecting metabolism, but at the same time, through JAK kinase, its primary mode of action on IGF-1 is through regulating transcription, which takes a lot longer, for example, than blocking glucose from the liver, but it does in the liver. The other things don't.
So technically they all alter gene expression?
I wouldn't say that. They all have the potential to. And many of them, like if you take Gary Firestone class, you'll see interact with more than one second messenger system. All you have to know from me are the ones that we focused on. It'll get more complicated as we go further in the course, But even from what we do is much more complicated than what we'll do in this course. Simply because my focus is on physiology, growth, and reproduction. So I'm giving you what you need to know to understand how hormones regulate those things. Okay. Other questions or comments? All right. So first we're going to talk about the radio immunoassay. And it was critical and still is in endocrinology in the field. Because hormones circulate at such low levels that it's difficult to evaluate them. You can't, estrogen circulates at picograms per mil. You can't extract and purify picograms of something and then just weigh it out and see how much it is. So people developed a technique for measuring such low concentrations, not just hormones, of anything, that might be circulating in the blood, or that you can extract and purify from tissue. We do it from tissue in my lab as well. And we're actually going to, Lisa, your GSI said, these assays are hard to understand unless you do them. We're actually going to do one here today. Sort of. Some years it works better than others. We'll see what happens. So it's called a radioimmunoassay. Assay just means test. Assay just means test. Radio, because what we're going to do is we're going to use radio-labeled hormone, which I'm going to color in orange. In the case of a steroid, for example, it's a steroid that's trigated usually at the 2, 4, 6, 7 position. You don't need to know that. It's immuno because we're going to use antibodies. Which I'm going to draw like that. And these are antibodies that have been raised to the hormones of interest. And the other component is the radioinert, or just regular old hormone. You can do this as long as you have the reagents, which can be expensive, as long as you have an antibody to your hormone of interest, and a radio-labeled copy, and a set of radioinert standards. You can do this for any hormone. In fact, you can do this for any compound that's at low levels, as long as you can purify it. In the case of steroids, we're often interested in measuring hormone levels in the plasma. So in the case of steroids, there's usually an extraction process from the plasma. You can tell me why that is. You typically don't have to do that for protein hormones. So here's what you do. You take a blood sample. You take the plasma. So you use something that separates the cells from the plasma, from the water particles. Why do you think you would have to do it? You do an organic extraction to get the hormone that you want to measure. What's your name?
Alvin.
Alvin.
Because they're in the cell.
They're not in the cell. Who's that back there? Yeah, so they're bound to protein. So you're trying to measure something, you can probably guess, you're going to try to measure the antibody. You have to get it away from the binding protein, right? Because if this is remember those amphipathic, whatever that word is. Amphipathic. Remember the hormone, if my hand is the binding protein, the hormone's hidden away now. You can't do anything to it. But if you extract, for example, let's say an ether, you unfold the protein, and now the hormone gets released into the organic solvent. All right? So you have to do an extraction process. And then what you want to know is you want to know how much hormone is in that unknown sample. Here's how you do that. You first have to generate what's called a standard curve. That means that you have purified forms of hormone of known quantities, and you're going to use that to kind of, let's say, make a ruler. We good? Here's how you're going to do that. First off, you're going to take a tube, and in that test tube, there's antibody. Let's put 3 of them in there. And then in that test tube, you're going to add the radioactive hormone. And the reality is you add more of the radioactive hormone than the antibody can bind. And then you do a procedure, it depends on what kind of hormone you're measuring, where you separate out the bound from the free radioactive hormone. And then you can measure, right? You can measure radioactivity. You can't see it and you can't weigh it, but you can measure radioactivity, right? And for tritium, you use something called a beta assimilation counter. And you'll get some level of radioactivity. That is a measure of how much antibodies bound to radioactivity. Are we good so far? Stop me if you're not, because if you're confused now, you'll be really confused in a few minutes. I'm going to make a number up. You get some number. Yeah, what's up? What's your name?
Sean.
What's up, Sean?
So why do you need to add more hormones and antibodies?
I guess probably because you don't want the antibody saturated. But you'll see how it works in a second.
But if you add more hormones, then the antibody saturated first.
Well, you'll see what I mean in a second.
Okay.
Not with the radio inert. Then the next two, what you're going to have, and building a standard curve. So this is your standard curve. The next tube, you're going to add a small quantity of radio inert hormone. So the same amount of antibody, same amount of radioactive, but now you add a little bit of radio inert. Next to we add a little more radio inert. Dot, 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 dot. All the tubes, all the tubes have the same amount of radioactivity, but you're increasing, let's say 10 tubes, the amount of radio inert of known quantities. So this is your standard, right? So by the time you get to the last tube, so if I plotted this on this graph, what would it look like? What's your name again? Up two. Two. And it's actually on a log scale. So the radio inert is competing for the radioactive. The more radio inert you put in, the less radioactivity can bind. So that's what I meant, Dan. You want the antibody to be limiting so that there's competition. Maybe I used the wrong term. What should the slope of the linear part of the curve be? Who has a guess? Okay, hi. What do you think the slope should be?
Dissociation
No, we're not there yet. No, what number should the slope be? You're skipping all the way to something else. What do you think the number should be for the slope? And it's a log scale, but for the linear part, so you have some low levels of radioinert that just can't compete with the radioactive. You reach a point where this becomes linear, and there's competition, and then you reach a point on the curve where there's so much radioinert that the radioactive can't compete. What should that slope be? If you had to guess, what would you say? What? Negative. Yeah, the slope should be 1. Absolute value should be 1, correct. What's the symbol for absolute value? Is it 2? Long time since I had that. Why should the slope be 1? Oh, just like that. That's what I was going to do. That's makes it right. Why should it be 1? Who has a guess? Because the affinity of the antibody for the radioactive and radioinert should be the same. Nanogram per nanogram. If it's not 1, then you know you have a problem in your assay. And sometimes it's not. If you have a heterologous assay, don't worry about that. But your slope should be 1. That's your standard curve. Now what we have, we're going to do what we're going to do this in a second. Now what you have is you have some unknown sample. You have somebody's blood plasma in your hand. And you want to know, for example, let's say we're doing progesterone. You want to know if they're pregnant. And a good way to know how pregnant they are is how much progesterone there is in their blood. So you have this standard curve where you use known quantities that compete against the radioactivity. Take your person. Take a blood sample. Take the plasma. That blood plasma is then extracted because the progesterone is bound to a binding protein. And then everything's in that plasma. You don't actually have to purify the steroid. Her progesterone's in there, her estrogen's in there, her testosterone's in there, her aldosterone, her glucocorticoids, all that's in there. But you then put in a tube with the same amount of antibody as in your standard, same amount of radioactivity. And then she has some unknown amount of endogenous hormone, of progesterone, in her blood. But in doing this assay, there's going to be the same competition. It doesn't matter the testosterone, estrogen, everything else in there, because the antibody, at least you hope, is specific for progesterone. And you'll get a number of how much radioactivity is bound to the antibody. You'll get a number that, let's say, is here. And then you use your standard curve to estimate how much hormone's in her blood. You can't see the radioinert hormone. All you can measure is the amount of displacement of the radioactivity from the antibody. So you'll get some number. And you have a question? You'll get some number of how much radioactivity is bound. And that allows you to estimate how much is in the blood. So the reality is, you can't measure it directly. It's an indirect measure. And your name's Andy.
Yeah.
Yeah.
So is the standard curve standard for everyone?
No, the standard curve is made from In this case, progesterone that you bought from Sigma Aldrich Chemicals.
Oh, okay.
So the standard curve is pure progesterone, and you're using antibody to make the curve, and you're assuming that the progesterone that's in your blood is the same as what's being measured. If not, if you did a dilution series, you wouldn't get a slope that was 1, because it wouldn't be competing 1-to-1. And so, for example, I work on frogs. Sometimes there are weird steroids in there that compete with the antibody, and then you don't know what you're actually measuring. So that's the drawback. Like we actually have this problem with my lab down there. There are other problems too, but for humans, it's pretty much we know all the steroids and all the things that are circulating, and it's pretty easy to figure out.
Okay.
For odd animals, it's a little more difficult. Dina, right?
Yes,
Okay.
Are human plasma levels pretty consistent across people?
Oh,
Or
absolutely
there-
not.
Okay, so
There's-
how do you control the fact that like someone might have more fat circulating in their blood than someone else, which would change their concentration per milliliter?
Well, it wouldn't change the concentration per milliliter,
Okay.
right? Because you're measuring against how much plasma there is. So it's not relevant. So there are other things that are in there, but still so the hormone, the concentrations are going to change time of the month, time of the day. There are racial differences in some hormone profiles. There's how much stress you have, whether or not you have a disease or a disorder. There are a number of reasons, but it shouldn't affect how it compares to the standard curve. And it's usually in humans, it's like nanograms per deciliter or something like that, but same idea. But that shouldn't change. It might change, for example, I know this is true in frogs. If you're dehydrated or your blood volume is low, it might artificially seem elevated. Which actually doesn't matter, right? Because if it's elevated, your body still reads it as a higher concentration. Yes? What's your name? Adam.
So when you're dealing with like hormones that are really similar, I guess like IGF-1 and insulin, because you have structurally, would it get increasingly complicated to find antibodies that would want?
Not for humans. You get antibodies for anything. In fact, there are antibodies that will distinguish between testosterone and DHT, which differ by 1 proton. There are antibodies that are distinguished between cortisol and cortisone, which differ by 1 proton. So you can do that. For humans, yeah. When you're working with weird animals that might have weird steroids and different amino acid sequences for their hormones, then it becomes more difficult. Like you have to generate your own antibodies. But for humans, it's you can measure anything you want. Yeah, and then we're gonna do one.
So what does this curve tell you? What does the curve tell you if it's less than the 40s?
Oh, if it's less than the 40. So it is possible, for example, that your hormone levels are so low. Is it possible that your hormone levels are so low that they're not on the linear part of the curve? That there's not enough hormone in your body to compete with the radiolabel stuff? Wait, who just asked me that question? What would you do in that case? If your hormone levels are so, I know you asked a different question, but if your hormone levels are so low and you're running a radial aminoassay, how could you try to get a better number? Because if it's not on the linear part of the curve, then you're going to get screwed. What? You could redo your curve, but there's usually a lower limit to the antibody. Like it can't go all the way down to like femtograms.
I was gonna say decrease the amount of radioactive.
You could do that, but there's still a limit into what absolutely, that'd be a great idea. But then there are other problems that generates an assay. What's one other thing you could do? Oh, you guys probably can't figure it out. So you can increase the volume that you're extracting, right? Because you control how much volume of blood that you're using. So you can increase the amount of volume to pick up more hormone, and then you just correct for that volume. So, but your question was, if you're on this end, if your hormone levels are so high that you're off the curve, right? That was your question. Like your testosterone levels are so high, you're way out here somewhere, you can't get accurate reading, then you can do the same thing. You either extract a smaller volume of blood, or after your extraction, you can dilute it so that you get back on the linear part of the curve. Okay? Now we're going to, yeah, I figured your name.
Sabrina.
Sabrina.
So even if you have something that's like so similar that it can bind to the same receptor.
Not receptor, antibody.
No, but like say you
Oh,
have two hormones that are so similar they bind to the same receptor.
Uh-huh.
Like, I believe insulin and IGF-1. Would
Yeah.
those still have like separate antibodies? Or?
Yeah. So, yeah, you can, like I said, there's separate antibodies that measure cortisol and cortisol, DHT and testosterone. The specificity for the receptor is unrelated to its ability to bind the receptor. Unrelated to its ability to bind the antibody. Because they're different episodes, they're different, they're binding to different. Okay, hi.
So what advantage of this assay over ELISA? Can we measure steroid using ELISA?
You can measure, we'll talk about ELISA later in the course. The pregnancy test that you take at home is basically an ELISA. So an ELISA is an enzyme-linked immunosorbent assay. And the only difference is, well, there's some that are direct, is that you're using a color change instead of radioactivity to measure. And so a lot of clinical labs and things like that have switched over to ELISAs. For those of us who do comparative biology, so the principle's the same. Just so you're measuring a change in color versus a change in radioactivity. For those of us who do comparative biology, most of the ELISAs don't work. Because they've been worked out for humans, or rats, but they don't work for people who are doing comparative
So
biology.
which assay would be more sensitive to the concentration?
I don't know that anyone's more sensitive. Radioimmunoassays vary in their sensitivity, and so do ELISAs. So some ELISAs are more sensitive than RIAs, but RIAs differ as well. Because like estradiol assay is notoriously difficult and not so sensitive. I don't know why. And impossible almost to measure in rodents. Very few people have done it reliably. I don't know why. I don't think anybody knows why. All right, so here's what we're gonna do. If you got a bag, then you're gonna help us do a radioimmunoassay. I hope this will make it clear. So if you have a bag, here's what I want you to do. Here's what's in your bag. What's in your bag are good and plenties. They're red ones and you do not eat them. They're like 10 years old at least. You have red ones and you have white ones. So the red ones are the radioactive ones. And you can only see the red ones. You can't see the white ones. Your hand is the antibody. And it can't see if you're going to shake your bag up. Don't do it yet. And don't shake it too hard, so it'll crack the like an ice, or you've been playing, whatever they are. So you're going to shake it up, it's a centrifuge. Your hand is the antibody. Your hand can't see if they're red or white. So what you're going to do is you're going to reach into your bag, when I tell you to, and you're going to pull out, without looking, you're going to pull out 10 good and plenties. If you have a number on your bag, that means you're part of the standard curve. Okay, so let's make our standard curve first. If you have, let me show you how to do this. I'm going to say, my bag says 5. That means that I have 5 whites in my bag. I know that because I made the bag. 5 means I have 5 whites in the bag. So my hand is the antibody. I'm going to pull out 10. And don't lose these, because then my GSIs have to count them all over again. So I pull out 10. 1, 2, 3. So I pull out 10 without looking, and I get 7 reds. All right, so if you have 0, that means that you're at this point. If you have 0, shake your bag, pull out 10, and tell me how many you get. This one always works real well. So if you have 0, pull out 10, tell me how many good and plenties you get.
Hurrah, 10 reds.
10 reds. Who else?
10 reds.
10 reds. We're going to take an average. So you replicated this. I'm going to guess the last is going to be 10. All right, so there we go. So this is your on the bottom x-axis is your increasing radioactivity. If you have 5, like I did, reach in and tell me how many you get. The same. Shake it up, pull out 10, and you can only count the reds. You can't see the whites. Yeah? 7? Oh, you're not pretty good. Who else? Who else had 5? How How many do you get?
3.
3? Bad point on the curve. So 7, 7, and 3 is 17 divided by 3 is like 6 point something. Did I do that right? 5 point something? Okay, if we get a bad point. Alright, if you have 10, what do you get? You can only count the reds. 4. The rest of you guys can go ahead too. So 4, what else you got?
6.
6. Oh, average of 5. What else we got? Our other point should have been somewhere over here. Where's my other 10? Is there another 10? Huh? What? Okay. 4 divided. The average is 5. We're good. All right. Except for that bad point. If you had 20, what did you get? 4. What else? 2. What else? Is that it? Does anybody else have 20? Huh? Did you have 20?
No, it's 2.
Patient 2. You're not part of the curve. So, okay, so that's it. So 4 and 2 is 3. I mean, 4 plus 2 divided by 2. You know what I mean. All right, if you got 40, what do you got? 2. 2, what else? No, that's all we have? Okay. Oh, you guys are like doing a bad radio. You know, that's it here. So more or less, it should look like that. All right, if you have patient 1, how many reds do you get? So this is our unknown.
1.
1? Yeah? What? None? 0? Is that it for patient 2? Wasn't there a third one? Oh, you guys are blowing it. Oh, no, patient 2. Sorry, patient 2 was how many? 1 and 2? I'm not doing this again. So patient 2, what were the numbers again for patient 2? Patient 2?
I got 7.
No, patient 2. What? 6, and what was the other number? 7. 6 and 7. So 6.5. Wait, were you patient 1 then?
I got 3.
6.5. So 6.5 is about here. I would guess if you count in your bag, there'd be 10 whites. So you count your whites and figure out. Patient 1 is how many?
1.
1? What else? Just 1? There's only one patient number 1 bag?
No. They're just not answering
1 and 0. Oh, you guys blew mine. So I would guess. So 1 and 0 is about 0.5. I would guess you have 40 in your bag for patient number 1. But anyway, you get the basic idea of how it works. You can't see the radio inert stuff. You guys have bad hands. Put the candies back in the bag. Last year's group did better than you guys. The guys, you can't see the white ones. You can't see these small amounts. So you're using the competitions for the red ones to determine how many white ones there are in the bag or in your test tube. Yes.
So when you mix in the labeled and unlabeled, do you do it at the same time, or is there one? So you must have to do it at the same time,
exactly
right?
The same time.
Yeah.
Correct. So well, that's not true. There are some assays where you pre-incubate and then you add in the radio inert stuff. But typically most assays are added and incubated at the same time. And I actually don't know why some have a pre-incubation. I have to get back to you now. But most are added at the same time. Other questions or comments? All right, put the candies back in the bag. Close the bags up. We need to collect those at the end of class. The next assay we're going to talk about works a little bit similar. The next assay is a radioreceptor assay. And in this case, you aren't looking for the hormone. You're trying to find out how much and what kind of receptor there is in a given tissue. This assay only works for soluble receptors. Who knows what I mean by that? This assay only works for receptors that are in the cytosol, or it could work for a binding protein as well. It cannot work for a membrane-bound receptor, although there is one paper that we found a couple years ago that used some kind of special radioreceptor assay for IGF-1. But in general, it only works for cytosolic receptors, for soluble receptors. And in this case, it's radio because we're going to use radioactive hormone. And the principle is very simple.